there are things that you can look at in terms of continuity of sleep as well. So how long do you sleep for? How long does it take you to get to sleep? How often do awakenings occur? How long do awakenings last? And what's your arousal threshold? How much of a stimulus does it take to wake you up? We tend to look at the microstructure of sleep. So we take the EEG signal and we decompose that into power spectral analysis using sort of Fourier analysis. We also look at something called the K-complex, which I'll be able to talk about in more detail. So the K-complex is really a single instance of one of these slow delta waves. They can be elicited by sounds. So here we've got auditory stimuli. We have people going to sleep with little inset earphones. We play them tones. They're quite loud. They're at 80 decibels, but they're very short, only 50 milliseconds. So they tend not to wake people up. Sometimes the K-complex will be elicited by a sound, and sometimes it won't. So you can again get an incidence measure of, is the brain in a good state to produce these slow delta waves? You can average them and get a better signal-to-noise ratio from the averaging, and you end up with this very nice, smooth waveform, and you can measure the amplitude, which gives you an idea of how many neurons are involved and how well synchronized they are. You can plot it across the head. In adults, it's always largest at the front of the head, and it gets progressively smaller as you move back. And this is another way of representing that. So this is a top-down view of the head. That would be the center of the head, left ear, right ear, nose, back of the head. So this is that most negative peak. And you can see it's very nicely symmetrical across the two sides of the head. It drops off front to back. And it really doesn't matter what type of stimulus you use to produce it, you end up with the same type of activity. In this case, we're using tones on the right there and blockages of breathing. So you come to sleep, you're wearing a mask, and every now and again we put our finger over the hose, essentially, and see what happens. We've been able to work out, sort of using something like seismography, to track back into the head to work out where it's coming from, and we think it's coming from very deep in the frontal cortex, and it spreads up and then comes back down again. So we use this as a tool, and one of the ways we use this as a tool is to look at sleep and normal aging. But now we have the audience participation part of the evening. You get to vote now. So I want a show of hands. How many of you sleep less than you did 20 years ago? This assumes you can remember 20 years ago. But okay. How many sleep more? Okay, so most are sleeping less. All right, good. So far, so good. How many of you are woken up more easily now than you were 20 years ago? I assume the waving means very much more easily. Okay, and how many of you are sleepy during the day? Okay, how many of you are more sleepy during the day than you were 20 years ago? Okay. See, that's not the way I wanted that to go. This is... So actually, there'll be cash remuneration for people who say less. What happens? Well, this is a hypnogram, and we can plot arousal state or sleep state where you have basically depth of sleep going down. The green is light non-REM sleep, and the blue is that slow wave sleep, and the red is REM. So in young adults, you have these periods of REM that get progressively longer across the night. They come about every 90 minutes. Most of the slow wave sleep is consolidated into the early part of the night, the first half of the night. And these little blips down here are arousals, very brief arousals, not really awakenings, just for a few seconds. Okay, and this is an elderly person. And again, my definition of elderly has also gone up. So this person is 135. And you can see there's much less, in fact, almost none of this slow wave sleep. The REM periods are still occurring, but they're shorter. There's periods of wakefulness now, these gaps here, and many more arousals. So a friend of mine who sort of invented behavioural therapy for insomnia, a guy called Dick Bootson, described the sleep in the elderly as brittle and fragile. it's harder to get. When you get it, it's harder to keep, and it's easily broken. Now, it gets better. As I'm discovering, as you get older, things fall off, things fall out, things stop working, and your brain is no exception to that. So aging is associated with a number of changes in the brain, a decrease in the cortical volume, so you lose neurons as you age, and a decrease in the integrity of the white matter. So the white matter are the wires that connect the different bits of the brain together. So not only are you losing bits, you're showing not huge in normal healthy aging, but nonetheless measurable degradation in how well things communicate. This is work by our human neuroimaging group here at SRI, headed by Dolf Pfefferbaum. And this is I don't know how many millions of dollars it took to make this slide, but this is the results of many studies where we look at individual data points. So these are just snapshots in time of many different people. These are 95 normal healthy men. There's another slide I could show of I think 70 normal healthy women. It looks exactly the same. That your cortical grey matter goes down as you get older, and it's replaced by cerebrospinal fluid. So at the bottom you've got an 18-year-old brain compared to a 76-year-old brain, and you can see that the fluid-filled spaces in the brain are much larger in the older brain than they are in the younger brain. This is now longitudinal data showing that not all parts of the brain age equally. So you can see here the prefrontal cortex, which is associated with a lot of the higher order cognitive processing, thought processing, etc. The stuff that doesn't develop till much later in young men than young women, for example, the bits that involve judgments and responsibility. So I'm still waiting for mine to kick in. That ages faster than other bits of the brain. And these are data from the same group. This is a joint group we have with Stanford Psychiatry, headed by Dr. Sullivan. And this is showing decreases in the integrity of white matter, again, over aging. And these are white matter pathways that are connecting the two halves of the brain in this instance. So we started testing hypothesis several years ago that this should lead to diminished ability to produce this very large slow wave EEG activity, because you need lots of batteries to produce a big voltage, and you need them all firing at the same time. So we did a study in a large number of young and old people and found a number of differences in sleep. So it took, if you look at the sleep onset latency, it took longer for the elderly subjects. These are people in their 70s compared to people in their 20s. Longer to go to sleep by about 20 minutes. There was more of the very light drowsiness, stage 1 sleep, much less of the slow wave sleep, so only 7% of the night versus 20% of the night, less of the REM sleep, less total sleep time, more time awake, and thus the sleep efficiency went down. So sleep efficiency is one of the ways that sleep researchers try to pretend that we're really hard scientists, because we come up with numbers. So you divide the number of minutes awake by the number of minutes in bed and come up with a percentage, and that's sleep efficiency. Okay, so the most important thing I think here, well, for us at this point, was the slow wave sleep change. And if slow wave sleep changed, we figured that K complex, the way we can experimentally elicit the delta waveforms, should change as well. So in the reddish colour here, the pinkish colour, we have the average evoked response from the young people, and here we've got the average evoked response from older people. And it's still there. It's still firing. It's just smaller. It's also less likely to occur. So we were only getting these K complexes to about 40% of stimuli in the older versus 74% of stimuli in the young people. Did a much larger study after I came to SRI. This is results of, this is showing the age distribution of over 80 subjects that we looked at. This is what happens to the K complex incidence. You see, it's just a very slow decline, but a lot of variability. Less variable is what happens to the amplitude. So it's a negative waveform. So moving back towards 0 means it's getting smaller. And you can see this very nice linear relationship associating age with the amplitude of the response. So if you plot these sort of decade by decade, this is what the response looks like in 19 to 30-year-olds, or as I now refer to them, children. Bigger at the front, getting progressively smaller. This is 31 to 40-year-olds, so quite a marked drop. 41 to 50-year-olds, not much change between 40 and 60, as it turns out. 61 to 70 is also, let's see if I can just go back, yes. 61 to 70 has this interesting thing. The amplitude doesn't change, but the latency does. So it gets slower. It takes longer for it to be elicited. And then 71 plus, it's later and smaller. Everybody feeling good now? And this is just plotting the amplitude as a function of where you are in the head, and you can see that the steepest slope is at the front of the head, and the biggest change is at the front of the head. And we've now got data because we've got MRI magnets here, and we can measure people's brains as well as measuring their sleep, so we can relate the sleep changes to the brain structural changes, and we've got reasonable data indicating that they're nicely related. So here we've got the lateral frontal grey matter volume correlated with the amplitude of the response. You can see a nice straight line there. It's interesting that even when you throw in a whole bunch of different brain regions, though, the age response, it's only explaining about 50% of the variance. So there are other things going on in the brain other than just getting old and just losing grey matter. The other thing I want to talk about aging is obstructive sleep apnea. I was really lucky to be part of the end of a study that really never got its due rewards. It was started by a guy called Don Bleiwise in the 70s when he was at Stanford. It's called the Bay Area Sleep Cohort. He enrolled over 100 people who were in their 60s, mean age 57.8 at first entry. They were healthy people. Okay, screened healthy people, so we checked them and made sure they were healthy. And then he looked at them at 5 and 10 year intervals, and I did the last batch here. So these are now people in their 80s, and we got 30 subjects. So this is the first 15, and we're able to plot over time what happened to their obstructive sleep apnea index in the blue lines, and what happened to their body mass index in the red lines. So we know that as people get older through normal adulthood, so you know from the range that sleep clinics normally look at, say 30 through to 60, the apnea index goes up, but you get heavier as well. So it's all thought to be related to body weight. So what we found was with looking at these much older people was that their apnea index went up. Almost everybody went up. Irrespective of whether their body mass went down, went up, or didn't change. So it's probably a loss of some muscle tone in the upper airway, some changes in neuromotor regulation of the upper airway, and so on that are going with aging as well. The manatee here is because you need cutesy animal photos, and the manatee is actually having an apnea. We know that because he's not dead. And he's underwater, and therefore, and asleep, so not breathing.